in the context of pain, so I will pay also attention a little bit to the anatomy, because that's, let's say, the fundamental, otherwise you cannot understand. So sometimes you see parts about the anatomy of the pain system, sometimes a little bit more in detail when necessary. As you can see here, the team are split, and maybe interesting for you. We are translational research. We do research, pain research in the clinic, and attached to that is the research in the laboratory. And we have quite some Belgian fellows who originated from Diepenbeek, Hasselt, for instance, Robbie Jager, who developed this thesis last year. Lisa Knappen, she's working now in my lab on her thesis, tries to finish that by this year. We have Sophie Janssen, a postdoc, who also worked here in Diepenbeek in Leuven, and then joined our group. And we have, of course, also a French-speaking Belgian PhD student, Anne Gabriel, who finished her thesis 2 years ago. And I will tell also a little bit about her work. So it will be a mixture of, let's say, literature and ongoing research in the laboratory. Now, if you talk about pain and inflammation, this is well known. You can have inflammation in the leg or the arm. And what happens? The signal here is transmitted to the dorsal root ganglion, that's the cell body, and enters the spinal cord, the central nervous system, here in the spinal cord, dorsal horn. There are pain neurons located, and these pain neurons, they further send the signal up to the brain. I will go into detail about the anatomy of the, let's say, the supraspinal projections. But let's first start with what happens here. Of course, you get inflammation in this area. And this is already a rather complex cartoon with a lot of information. After pinprick, for instance, you can get inflammation. What happens with inflammation? You see distortion of the skin, and a lot of factors are released, from substance P to bradykinin, serotonergic molecules. And they all, and this is the one, the very important one, cyclooxygenase COX-1, COX-2. A lot of therapies have been related to modulation. This enzyme, this COX enzyme, who further downstream activates prostaglandins. And these prostaglandins are known to affect on the blood vessels. And here you can see swollen blood vessels give redness, the white blood cells escape from the blood vessel, and there is leakage which can give edema. So this is an important target in pain research. This is, in fact, the peripheral aspect of pain, the peripheral sensitization, what happens. Another important mediator are the free radicals. Kia Hageman told me to pay a little bit attention to the free radicals. They are in balance with antioxidants. They are very short-living oxygen radicals who can be normally released from the cell or from the extracellular area by antioxidants like vitamin E or glutathione. Now, if there is damage, the balance is no longer optimal and you will have oxidative damage. These radicals can damage proteins, and that in itself induces inflammation as well as tissue injury, and then you see the whole cascade with the prostaglandins, as I showed you in the previous. So what are these free radicals? This is a nice cartoon. They are like robots, which are deficient in energy, and they attack the other molecules and snatch their energy. And here you see an example of vitamin E rich food who has an antioxidant behavior. these free radicals, they attack the membrane, and finally that results in death of the cell. And that in itself releases lots of molecules, inflammatory molecules. Now, inflammation and oxidative stress and pain. Now we go to pain. What happens with pain? In the skin, you have endings, nerve endings, blunt nerve endings with a lot of receptors. Here you see these transient receptor potentials, ion channels. You see a lot of them here, TRP, V1, V3, V3, V2, and A. They are opening upon this inflammatory response in the skin, and you can see that finally results in a generation of a lot of action potential. So these blunt endings, characterized by presence of a lot of channels, induces axipotentials, and these axipotentials will be sent to the spinal cord. Now these endings of these pain fibers are characterized by these channels, cation channels, present here, and you can define them. For instance, you have the endings with a majority of TRPV1 receptors. They are known to respond to hot peppers, capsaicin. There are other ones, and you see here indicated when they are activated. You see there are some, they are more for the cold allodynia, for the cold response. Based on these, the presence of these receptors, these C fiber endings, pain fiber endings are characterized, identified. So you have a lot of possibilities. And finally, the signal from these afferents, from these pain afferents with the TRPV receptors and the axipotentials enter the spinal cord, and what happens here? Here, and as you can see it's indicated the pain gate. At the pain gate in the central nervous system, it is decided whether the signal will be sent further or not. It's a very important place. But before we go and talk a little bit more in detail about the pain gate, here about the anatomy. As you know, the spinal cord, the transverse section, contains a lot of fiber systems. Descending fiber systems in the rat. This is the corticospinal tract, in the dorsal horn, whereas in the human, it's in the lateral, here is the ribrospinal tract. And in red are ascending systems. For instance, here is situated the spinothalamic tract. The spinothalamic tract contains the pain fibers. So from the pain neurons in the dorsal horn, the signal is ascended through the spinal thalamic tract up to the thalamus, and from there to the brain. This is another important player in the pain signal, the dorsal columns. They are situated, of course, in the dorsal funiculus. If you talk about pain, I included this and two more slides because I think it's important to realize that we are not talking, if talking about pain, not exclusively about the central nervous systems, of course the sympathetic and the parasympathetic nervous system involved in. But here you see a part. Here you see the truncular ganglion sympathetic connected with the spinal cord. And you see here the spinal cord again. Here are the dorsal root ganglion. So the afferent fibers, the pain fibers, come in here via the dorsal root ganglion and enter the dorsal gray matter in the spinal cord. And then, as I showed you, they will be transported. The signal will be transported back to the brain. There is also afferent output, you can see from motor neurons, through this same nerve, spinal nerve, and connecting with the sympathetic ganglion. And I guess you all know this type of cartoon, and it is important to understand. Here are, of course, here is the spinal cord connected with the spinal ganglia or sympathetic ganglia, and they are very important in regulation of all the viscera. And on the other hand, you have the parasympathetic system releasing from hypothalamus, for instance, factors which also attack these viscera. So if you talk about pain, you have a signal coming into the central nervous system with an immediate connection with the sympathetic nervous system, which regulates the viscera. Please remember that. Now, if you then stay with the central nervous system in the spinal cord can be subdivided anatomically. And here you can see it's been subdivided based on the morphological characteristics. For instance, here there are large neurons, motor neurons that's designed Rexit qualified that and said this is area 9. And he started from here, layer 1 and 2, one on top, 2, 3, 4, 5, and so on. And the pain fibers, they enter the dorsal aspect of the spinal cord and terminate mainly in these layers here, in the upper dorsal layers, 1, 2, and 3. There are the pain neurons. Here again, you see these, again, important to know is, because later on I will talk a little bit about animal models where you make a peripheral injury, that input, afferent input, not stays within one segment. You can see here tranches go to several segments. So you can, for instance, make a lesion in this one that induces inflammation and gives a pain response, whereas the other ones are intact and allows the animal to retract or to respond to this pain signal. Is that clear? So anyway, if you have questions, please ask them now or maybe at the end of the presentation. Now if you go to the afferents, you can see here 3 types of afferents. You have this unmyelinated C fibers, also entering, they are all entering this dorsal area. There are A delta fibers, thinly myelinated fibers, and A beta fibers. Now these A delta fibers, as you can see here, with a hammer, if you hit it, they give a very quick pain response. And the C fibers, the unmyelinated fibers, they also give a pain response, but a little bit later. The secondary pain response. And these A beta fibers, they are mainly for mechanical stimuli. So pressure on the skin is registered by A beta fibers. And they all project to the spinal dorsal horn, and here are the pain neurons, and you can see here already, again, this subdivision, these pain neurons, they transverse the spinal cord and then go up to the brain, the spinal thalamic tract. Whereas the others, and that's here, they enter the dorsal columns. So there are 2 important systems if you talk about pain. 2 important ascending systems. One is this one, shown in blue. You see the pain fibers coming in, the C fibers and the A delta fibers mainly, in the spinal cord, and they grow up through the spinal thalamic tract and here in the thalamus, and then you have projections to the cortex. On the other hand, in red, you see these thick fibers, the A beta fibers, they also enter the spinal cord, do not cross at that level. They have a projection to the nucleus cuneatus, or the gracile, and cross at medullary levels, and then go to the spinal, to the thalamus, and then to the cortex. So we have 2 different systems. One is really for the pain, acute, fast pain of A delta. And let's say a little bit slower pain mediated by the C fibers going via the spinothalamic tract to the brain. And you have the more pressure-related pain via the A beta fibers, then going through the nucleus cuneatus here in the dorsal column. These fibers stay here in the white matter, go up to the nucleus cuneatus, and then cross medial lemniscus system, also called the dorsal column medial lemniscus system, versus the spinothalamic tract. Here again, a summary of the two: proprioception and touch by the dorsal column medial lemniscus system, starting from the A beta fibers, and the spinothalamic tract with pain and temperature sensations. Here, once again, the dorsal medial lemniscus system. Here you see these fibers coming in, not crossing in the dorsal column, ending in the nucleus cuneatus, then crossing up. And then, of course, the other one is the spinothalamic tract. Located here, you can see here more in the lateral spinal cord, whereas these thick fibers enter the dorsal column, so are located in the dorsal medial area of the spinal cord. Okay, that was a bit about the anatomy, general anatomy. Fibers come in, are triggered, as I showed you here, in the periphery, sending a lot of action potentials, and these action potentials arrive here on these pain neurons. And what happens in this case? Here again, you see, and here is in fact a subdivision in two, but it doesn't matter. You have the small cells, the C fibers. So C fibers come in, you have the dorsal root ganglion cells, and these fibers enter. And you have the thick ones, A betas, large cells also entering this area. And here you see at several levels the pain transmission neurons. And either projection through the dorsal columns or crossing via the spinothalamic. What happens at that gate? There seems to be an interaction between A beta fibers and the C and A delta fibers. So these two components, let's say the pain and the touch, interact. And it's called the gate control theory. So here I have pain fibers coming in, here you have the thick A beta fibers coming in, and here is the gate. This person tells, okay, not or a go or a no-go. And this is a simplified summary of what happens in the spinal cord. Here you have these thick fibers, and here you have the thin fibers. And these thick fibers project on GABAergic cells, as well as the C fibers. And these GABAergic cells, a huge amount of these cells are located in layer 2, layer 3 of the dorsal horn. These cells modulate the transmission either from the C fibers or from the A beta fibers. And you can see here, because here is a positive interaction, activation of the A beta minimizes, let's say, the transmission here, but also minimizes the transmission of the pain fibers. Presynaptic. As you may remember from your self-experience, if you have pain and you scrub a little bit, this pain is minimalized. So in fact, the touch and the pressure here minimizes the pain response due to this idea. A beta fibers interact via GABAergic cells with C fibers, the pain fibers, and the A delta fibers. Now, I pointed out here central sensitization, because the pain fiber comes in here, and as you may remember, a lot of action potentials come in, and there is of course a huge release of transmitters here. These are glutamatergic synapses. So glutamate is released, and here is a receptor. And one of the receptors for glutamate is NMDA. Did you ever hear about these receptors? NMDA and AMPA? Yes? So if you have pain, the question is what happens at this glutamatergic synapse? There is a sensitization called central sensitization. This synapse is becoming more sensitive, it's more effective in fact. What results in a continuous pain response and might result in a transition of acute to chronic pain. Why do people develop chronic pain? Might be related to the sensitization of this particular synapse. And as you can see here is a sign, presynaptic, postsynaptic. It is of course an interaction between the neurotransmitter and the receptor. And I show you a cartoon. This is not the one I want to discuss in detail. I want to discuss another cartoon in which I show you one aspect, the early onset, so the immediate sensitization of this synapse, which is transcription independent. And of course you have also a late onset, a transcription dependent, which shows longer lasting effects. So there's a very quick response upon the incoming action potentials, which changes the synaptic efficacy within seconds. And this is the cartoon I would like to discuss a little bit in more detail because it shows of an elegant way the interaction between the various receptors and the various molecules. First of all, one important thing. Afferents can release a neurotransmitter, but at the same time other molecules. It's important to recognize that. Also substance P, or does somebody know what this is? Lina? Never talked about nerve growth factor or brain-derived neurotrophic factor. Trophic factors important in plasticity of the system, inducing sprouting. So they are also released by the same target. Now this is such a C fiber releasing glutamate. This glutamate, of course, binds to a glutamatergic receptor, for instance, the AMPA. It's an ion channel. Upon binding, it opens, and calcium streams into the cell. And this has a consequence. Secondary messengers are activated, and depolarization of this postsynaptic membrane is in fact results. This depolarization results in a removal of the block of this NMDA. NMDA is blocked by magnesium, and the NMDA is also open. So when the NMDA is open and it binds with glutamate, then also calcium can stream into the cell. So then you have a very fast and huge amount of calcium streaming into the cell and giving postsynaptic responses. These AMPAs, they are recruited intracellularly, and let's say there is an accumulation of AMPA receptors in the membrane very fast, which also increases the efficacy of this synapse. So you have two aspects related to glutamate. One is an increasing amount of AMPA receptors, and the other one is due to the depolarization, the block of the NMDA removes and it opens. So then you have really a bunch of glutamatergic receptors active and open. So if you talk about central sensitization, which is considered to be the general principle underlying pain and chronicity of pain, it's related to the glutamate and the glutamatergic receptors. Then the key mechanisms are trafficking of these receptors to the membrane, the AMPA receptor, a depolarization which results in the removal of the magnesium, and alterations in ion channels and receptors activity due to post-translational processing, so the late onset sensitization aspect. I will show you that in a minute. Here again, the various steps. I mentioned them already. release of molecules like glutamate, neuropeptides, BDNF, but here I'm talking about glutamate, act on receptors, recruitment of these umpire receptors, removal of the magnesium block, and then finally phosphorylation of these NMDA receptors, which leaves these receptors open for a very long time. Lowering of threshold, other channels involved, calcium channels, sodium potassium channels, and then it all results in an increased excitability of the neuron, increased sensitivity. Now for the clinicians, and as we do translational research, it's important to understand such a mechanism that you want to use it. Can we modulate this central sensitization in the clinic? And by that, we think pharmaca needs to be developed, like NMDA receptor antagonists. And they have indeed been proven to be effective in neuropathic pain, in chronic pain of humans. Here you see various examples of this. Here you see the NMDA channel. You can block the channel by competitive antagonists. So related to glutamate, here is binding at the same site, so on where glutamate is. There's also glycine. There's also glycine site on this NMDA receptor. A lot of antagonists related to the glycine have been developed. You have, of course, uncompetitive antagonists, blockers. Instead of the magnesium, you can use another blocker, which closes the channel. And you have non-competitive agonists, which bind on a so-called allosteric effect. So there are a lot of options to modulate this NMDA receptor. But you might know that the NMDA receptor is not unique, because glutamate is all over the brain. Of course, it's important here in these afferents, the pain afferents coming in, but it's of course, it's for instance known, it's also important in learning and behavior. If you go to the hippocampus, or if you go to the cortex and you study it by presence of neurotransmitters, you will see that a lot of them, a lot of the neurotransmitter is glutamatergic. So if you then modulate this NMDA receptor, you will modulate a lot of processes, not only pain processes. But anyway, here again, competitive antagonists are used for treatment of chronic pain. Competitive antagonists blocking the glycine, or non-competitive antagonists either. For instance, ketamine. I'll show you later on a little bit more about what we do with ketamine. But in view of the huge presence of glutamate all over the brain, and the presence of the NMDA receptor all over the brain, and the NMDA receptor is involved in central sensitization, but it is also known to be involved in long-term, let's say, memory, you want to develop as selective as possible. And is it possible? The question is, these NMDA subunits, and a lot of research during the last 10 years has been focused on the subunits of the NMDA. They differ. Here you see the NMDA channel consists of 4 components. The number one subunit, 2 of them, and then there is a variability between 2, A, B, C, or D. And it seems now that especially the 2D receptor unit is selectively present in the area where the pain fibers come in. This is a localization study by immunocytochemical techniques. Here you see again a part of the spinal cord. Here is the dorsal gray matter. Here are the dorsal columns. And as I told you, fibers came in here and end here on the pain transmission unit. Now here you see, after a lesion, an induction of antibody staining, antibodies directed against the NR2B showing an increased presence of the NR2B. This NR2B is exclusively, almost exclusively present in this area. And because of that, it becomes a very important target for selectively modulating the pain without a lot of side effects. Before I go on, I just want to show you how we measure pain in the animals, so in the experimental setting. And it is, of course, much more difficult than it is in the clinic. I cannot ask a rat or a mouse or whatever animal, do you feel pain and how much pain do you feel? Does somebody know how we record pain in the clinic?
Questionnaire.
A questionnaire? Is it a scale we use, or is it just a questionnaire, or a happy feeling questionnaire, or whatever? In the clinic, we use the VAS scale, the Visual Analog Scale. It's a scale from 0 to 10. When people are asked, could you indicate on this scale how much pain do you have? 0 is no pain, 10 is unbearable pain. And it's been considered substantial if you score more than 4. So under 4, the doctors will say, okay, this is not enough for treatment or starting of treatment, but if you're over 4, say to 6, 7, doctors will try to help you with pharmacological information. Now, we can't ask that to our animals. And what we do with our animals is we have a kind of a scale based on mechanical anhedonia, which means increased response to pressure. An animal which has pain will retract its paw to filaments with a much less target force. For instance, with a target force of 0.6 grams. A filament number 7, it will respond, whereas it's normal, it will not respond to such a filament. And we consider that an indication of pain. In fact, it's mechanical related pain, mechanical anhedonia. So we test by approaching the filaments to the hind paw whether the hind paw will retract or not. In addition to that, we developed together with a company in Holland another test system, a more objective test system, we think. It's called the catwalk. Should have been named the rat walk, but you can see we put animals on a glass plate. Here's the glass plate. And light is sent into the glass plate. And when the animal crosses the glass plate, at the place where it positions the paw, there's a breaking of the light, and that can be recorded through the mirror by the CCD camera, and this one's attached to the personal computer. So you can start recording when the animal crosses the glass plate. This is from under the plate. And you can see here the different paws. So you have an objective measurement and you have a dynamic aspect because it records a whole run. The run is about 70 centimeters. Here you have an idea how the rat will feel, or the mouse, when it enters the catwalk. And often we reward them with a treat, with a small treat, when it adequately crosses the catwalk. And then finally, it results in recordings like this on the computer. And here you see, this is the time a paw has been positioned, or it's not positioned on the glass plate, so it's in a swing phase. You have stand faces, swing faces. You have paw print intensities, and all kinds of things can be measured. And you can easily imagine that a rat which does not comfortably feel with his hind paw will have will not comfortably, which not comfortably uses its hind paw, will not use it as much as it did before. So it will have longer swing phases, shorter stand phases, for instance. And we can record that with the use of this method. Here you see this. Here you see, this is pre-operative, and this is with this von Frey technique, as I showed you before. Let's say the conventional test for pain in animal. You see there's an increase. Or sorry, this is the von Frey, there's a decrease. It becomes more sensitive. It responds now to 30% of the grams it responded before the injury. And this is the weeks, so finally it disappears. You can see here with the catwalk we have mean intensity. What the intensity, let me see here. This is the stance or the swing phase, increased swing phases, as I told you, decreased stand phases, and intensity of the paw plate also becomes less. So you can use these parameters with the catwalk just by allowing the rat to run across a glass plate for pain assessment. And that's what we do at the moment. So a couple of these tests and the results from that will come back. For instance, in this one, here's another model. The peripheral nervous lesion, you get a huge amount of action potentials of these C fibers, of the A beta fibers. And the rats will result in retraction. They become more sensitive. This is the percentage of antinociception. Don't bother about phase 1 or phase 2. And here, what they did is they have used selective NR2B antagonists. And by using this, you see once, on the one hand, you see a dose-dependent increase in antinociception, so the drugs do work on pain in these animals. Animals become more antinociceptive, anti-pain, so they feel less pain. And as you can see, it's been compared with a anti-photosensitive calcium channel. This is the side effects. With low amounts, this calcium channel blocker will show side effects, whereas you don't see it with these selective NR2B antagonists. So you have pain with an effect, you have not the side effects. This is on a kind of rotor rod motor impairment. So does this animal have less motor function? You can go on, and here you see again antinociception, in this case tested with the von Frey technique. And you see the use of these NR2B antagonists very selectively minimize the pain in these animals. Now this is another example. I will skip that one. I go to electrophysiological. Let's say evidence for the effect of these NR2B antagonists. This is another one, selective NR2B antagonist. And here you see C fibers. You have a stimulus and you see a high frequency stimulation. This is the normal response you see. This is when you block this, let's say, this synapse, glutamatergic NMDA NR2B subunit containing synapse with this drug. And you see no longer this high frequency stimulation. So also at the electrophysiological level, as well as the behavioral level, as well as immunocytochemically, these NR2Bs are quite promising. And at the moment, in these years, you can see, and this is a publication from last year, people are using and testing these drugs in the laboratories. Here you have the ketamine. I showed you a non-selective antagonist of the NMDA. It blocks the NMDA receptor. It has an effect. This is again the force, so this is the von Frey. You can see here these animals, and when you use the ketamine, the treatment, they are no longer very sensitive. Here they are very sensitive. So to, let's say, to 0.1, 0.01 grams, they respond by withdrawing the hypo. And if you use ketamine, so you block the NMDA receptor, you see they are almost normal for quite a long time. This is weeks. But if you use taxoprodil, which is an NR2B antagonist, you have the same effect. And now I come to the, let's say, the side effects, and they tested that with a catwalk. The taxoprodil did not show the side effects as shown with norketamine or with, for instance, norketamine or with ketamine. These are other catwalk gait analysis related motor aspects, walking aspects. So these drugs selectively block the NR2B, have an effect, as I showed you, pharmacologically and physiologically on the central sensitization, and they are now in use of modulating this process. Molecules like drugs like ethanol, taxoprodil. There's a huge interest of pharmacological companies on these drugs because they don't show the side effects as shown with ketamine and the more non-competitive drugs. So here again, the cartoon. The central sensitization in pain. Glutamate, AMPA, recruitment of AMPA, calcium in-stream, deblock of the NMDA, phosphorylation of this NR2B, for instance, opening of this channel, and effects. It's more sensitive. The synapse does not need so many axial potentials to have, let's say, an activation of the postsynaptic membrane. You don't need that much glutamate as you did before because you have a lot of channels open. Of course, this has also a late onset, a long-term effect, and then these effects, which are started with secondary damage, secondary messengers, enter the nucleus, and through activation of MAP kinases and CREB, you will have long-term effects because new molecules will be generated. Molecules like COX-2 or interleukins, cytokines, and all kind of stuff is being released. And it gives, in fact, it minimizes to some extent what I showed you at the peripheral. So you have a peripheral sensitization with these molecules, COX, prostaglandins, and here again, in the central nervous system, you have release again of COX-2 and cytokines. It also gives a kind of inflammation, a central inflammation. And I will go into detail on this central inflammation. And if you talk about central inflammation, other cells become involved. Long time, people focused on, let's say, the neuron-centric view. So we have these afferents, you have these various fibers, you have the pain transmission neurons, you go through the spinothalamic tract, and you go to neurons in the thalamus, you go to neurons in the cortex, and they project back. It's all neurons. Central sensitization is in fact started, focused on the neurotransmitter release and interaction between neurons. Now people are moving a little bit more to the neuroglial view. Glial cells, like microglial cells or astroglial cells. let's say, the immune cells of the central nervous system, like the mast cells, or the Schwann cells, or the fibroblasts in the periphery, you also have, let's say, kind of inflammatory cells in the central nervous system, the glial cells, the microglial and the astroglial cells. And we did quite a lot of research on that. Here again, you see the fibers coming in, the thick fibers going in the dorsal column, the thin fibers coming in the dorsal horn, and then project to other fibers, and finally go through the spinothalamic tract. There's a whole network. It's much more complicated than the gate control in itself. I showed you the interaction of the GABAergic cells modulating, let's say, presynaptically the release of the thick fibers as well as from the thin fibers. But in between here, very small, the glial cell. And this glial cell becomes more and more and more important in the whole process of pain generation. It started about 2000, 2001 on a couple of very interesting reviews from a group in Boulder, the United States, who reported spinal cord glia are they new players in pain or driving forces for pathological pain. And if you now look in PubMed, you see a huge amount of publications related to the role of glia. And here you see these cells. Microglial cells, very small cells, monitoring the area. In the dorsal horn, of course they are all over, but we are now talking about where the pain fibers come in, and the microglia are of course there and modulating. And you have the astrocytes, the more star-formed cells, glial cells. And if we now go in and study in detail what happens with these microglial cells as well as astroglial cells, if there is pain, we see the following. This is a transverse section of the spinal cord. This is the gray matter, this is the dorsal horn. This is where the pain fibers enter. And you see all the small dots are microglial cells. You can go into detail. Here you see the cells. They cover almost the whole area. And what happens if you make an injury in the skin or in the peripheral nerve? This is even higher magnification. And now if you give a pathological trigger, you can see these cells become activated. Thick. They swell up. They are like bombs. And they're going to release a lot of factors. And these factors, these, let's say, inflammatory-like factors, are important in processes like central sensitization. They affect the central sensitization. In this cartoon, here you see this, let's say this is a pain neuron, and here you see the resting microglial cell, which becomes activated. And I told you these small ones, and going to release a lot of molecules. One of them in blue, a neurotrophic factor, brain-derived neurotrophic factor. These microglial cells respond immediately. If you make a lesion, it's a matter of minutes, and these cells swell up and go release a lot of molecules, including BDNF and cytokines. They also activate the astroglial cells. They communicate with the astroglial cells. The astroglial cells do not respond that fast. They depend on the microglial cells. So the microglial cell releases factors, which eventually activates the astrocytes. And so the concept now is that for initial pain responses, the microglial cells are very important. So the onset of pain, the onset of the central sensitization. Whereas the astroglial cells, they are very important in the chronification of pain, the transition of acute to chronic pain. So the first stage is the activation of microglial cells. They release chemokines, They present toll-like receptors, and they release ATP. Or they do not release, sorry, excuse me. They do have purinergic receptors which recognize ATP, and this ATP is released by the afferent. And this is rather complicated, but it's in fact the state of art at the moment. If you look at the microglial cell, they are depicted here. They present these purinergic receptors, P2X7, P2X4. You have two groups of purinergic receptors, the P2X and the P2Y. These are ion channels, these are G proteins. If you talk about the activated microglial cells with the purinergic receptors, which are able to recognize the ATP. ATP, this is the afferent again. Here you see the afferent again. ATP glutamate, but you also can release BDNF, a lot of factors, but also ATP. And this ATP is recognized by the membrane itself, presynaptically, but also by the glial cell. It's released instantaneously. And the glial cell binds the ATP through these purinergic receptors. And what happens is here you see in blue is the ATP released by this sensory after this pain afferent, recognized by the microglial cell. This is the microglial cell. Here are the purinergic receptors. They get open, calcium comes in, activation of MAP kinases, and a huge release of, for instance, BDNF. These microglial cells, they produce cytokines, BDNF. And why is this BDNF so important? This BDNF is important because it affects central sensitization. So here again, you have the ATP, bound by the P2X4, activation of MAP kinases, release of BDNF, and that affects central sensitization. And then I come back to the cartoon I showed you a couple of times now. And here you see this BDNF, which can be released by the afferents, binds with a receptor, and through an intracellular molecule, SARC, it can affect the central sensitization, the NMDA receptor. But now we have also a source, a huge source of BDNF by the activated microglial cells. They also release huge amounts of BDNF, which affects this process through this same pathway. So the cascade, we think at the moment, important for the onset of pain is ATP, P2X4, MAP kinases, release of BDNF of these microglial cells, and interference with the central sensitization. Okay, this is a summary of the various receptors tested. Here you have a gene knockout, antisense, siRNA, selective antagonist for the P2X4, the microglia, and the P2X7 microglia with its function, BDNF release, MAP kinases. And you see some, at some points the table is filled with anti-hyperalgesia, so anti-pain effect, shown in the gene knockout for these receptors, and sometimes we don't have the results at the moment yet. So not a lot needs to be done. You can block these purinergic receptors. Of course, pharmaceutical industries are interested. How can we modulate this central sensitization through deactivating the microglial cells? And here you see by TMP-ATP, which blocks these receptors with low affinity, and it has also some action on the P2X4. So it's not very selective. We do not have really selective blockers of these purinergic receptors on the microglial cells. But still, if you use this, and here you see again the von Frey score. If you make a lesion, you see these animals become very sensitive. They respond to filaments of less than 5 grams. And if you apply intrathecally the TMP-ATP, so let's say the P2X4 blocker, you see they do have less pain. And then it disappears. This is the presence of P2X4 in the spinal cord, and this is effect on pain itself. So there are, at the moment, blockers for these purinergic receptors to block the onset of pain, but they are not very selective. And in this diagram, you see a lot of information, but the bottom line is that we have a lot of openings in here. You see here selective blockers for the TMP-ATP, for the P2X4, and in which models it has been tested. You see inflammatory models, where they apply formalin or carrageenan into a nerve. You have neuropathic models, where they lesion the spinal of the peripheral nerve. Very models there are. And you have acute pain models, where you drop the paw into, let's say, very warm or very cold water. And if you consider all the models, you see that only for a few they have shown a high effectivity. For instance, for the P2X4, for this compound, it's yet at the moment shown high effectivity in a carrageenan like inflammatory pain model. Medium effect in the neuropathic models. So not very, very good. From these we don't know yet. But of course, people try to develop these very selective purinergic receptor blockers. But we are not yet there. Let me say that. Okay. First stage, onset. So the inflammation. First stage is the microglial activation of these cells. They swell up, become like bombs, and release BDNF, which interferes with the central sensitization process. And what happens then? Astroclear becomes activated. And then, of course, the research question is, how do they become activated? Now, first of all, here, a picture of the astroclear cell. Here again, transverse section of the spinal cord. You see here, this is the area where the pain fibers enter. We see huge amount of astroglial cells in a normal spinal cord. And we go to a higher magnification, you see they do have a different morphology. They are more stark. So this is supporting cells of the nerves. They are all over making contact. And here again, if you then lesion a peripheral nerve, you see that these cells swell up. Here again, you see they become thicker, hypertrophic. They swell up after an injury. But this takes place 7 days after an injury. Whereas the microglial cells is a couple of minutes, in fact, hours when we did these stages. So what activates these astrocytes? So is there a factor released by the microglial cells which tells the astrocytes, go on, react, and make molecules which interfere with these central sensitization process? Which molecule is released by the microglial cells? A Japanese group in 2008 discovered that it's interleukin 18. Interleukin 18 is released by microglial cells. Now, first of all, they did a staining for interleukin 18. And as usual, they focused on the spinal cord dorsal horn, and here you see 1 day after a lesion. They make this kind of lesion, so they make a lesion in the spinal nerve, including the afferents, including the pain afferents, which enter the spine. And if you then do that, and you look at interleukin 18, you see at 7 days, they quantify it at 3 to 7 days, they see a peak of interleukin 18. Of course, that's interesting. But is this related to microglia? And then they do double stainings. This is the interleukin 18 in the dorsal horn. This is a marker for neurons. And you see there's no overlap. This is interleukin 18. This is MAP kinase, marker for dendrites and axons. And you see no overlap. This is interleukin 18 with a marker for astroglial cells, no overlap. But when they were staining for the IBA1, for the microglia, they saw an overlap. So these microglial cells produce the interleukin 18. Oh. I probably left one, but okay. The same authors looked at the receptor for the interleukin 18, and they were able to show that this receptor is exclusively present on the astrocytes. And then they blocked this interleukin 18 receptor, and they were shown this to have an effect on the behavior. So in a summary, these people showed that the interleukin 18 is very important for, let's say, activation of the astroglial cells. It's been produced by the microglial cells, and the microglial cells communicate with the astroglial cells via this interleukin 18. Now, then the astroglial cells become activated. And why are these astroglial cells so important for chronic pain, for the chronicity of pain? And that's summarized here. Here you see the afferents again, with glutamate, the AMPA, and an MPA receptor. That's what we know. These astrocytes become activated with interleukin 18, released from the microglial cells. And they have two important characteristics. First of all, they release interleukin 1 beta, and this molecule directly interferes with this process. But they are in a shape that they continuously release this interleukin 1 beta, because they at the same time they release a trophic factor, and they have a kind of autocrinic loop which keeps them activated. So this BFGF they release activates the microglial cell of the astroglial cell itself, and this release of, and then it releases interleukin 1 beta, which interferes with the central sensitization. So the astrocytes stay activated to the release of this molecule. So if you summarize, let's say, the immune response in the central nervous system, you can discern two steps. A first stage on the onset, microglia, and a second stage, important for maintenance and chronification of pain, it's the astroglial activation. I showed you ATP release, purinergic receptors with not yet very selective blockers, activation of MAP kinase and release of BDNF, which affects the central sensitization, production of interleukin 18, which activates the astroglial cells, and these astroglial cells, they continuously produce a cytokine, interleukin 1 beta, which also interferes with the NMDA through this autocrine loop induced by the BFGF production. And then we have done a lot of experimental work. Many animals died already. And we have interesting ideas about microglia, purinergic receptors, BFGF, BDNF, but not yet resulted into clinical applicable therapies. So why is that? Why do we all this research and insight into the mechanism, and why do we not yet are we not yet able to use it in the clinic? There are a couple of reasons for that. I showed you details of the immune response, the microglia and the astroglial response. So it differs in place and in time, especially in time. Microglia early, astroglial late. But this immune response is also protective. Known to be protective. It's all not always necessarily bad and destructing things. I showed you the destructive part of the inflammatory, the central inflammatory response, but you also have neuroprotective aspects, and that differs. So it's a very highly coordinated response. Makes it very difficult to intervene with it in a correct way. Then there is another aspect. The astrocytes we study in the animals are not identical to the ones in the human. In the human, it's more complex. It's not microglia and astroglia, but microglia type 1, microglia type Z, microglia, and astroglia. So you have different types of subtypes of astrocytes in a human. And this I always include, or often include in presentations, just to give you an idea. If you talk about one astrocyte, I showed you the picture, thousands of astrocytes in one section of the spinal cord. Let's say hundreds of astrocytes in our pain area. One astrocyte, it's a human astrocyte in this case, has been estimated to contain as many as 2 billion synapses. We are just talking about the astrocyte. I showed you the microglia cell, I showed you the not only animals, so that, of course, the neuronal interaction. Just to give you an idea how complex this is. But of course, as researchers in mass rate, we go on. And we talked, and we thought, it's such a complex and such a difficult system. Of course, I think pharmacological tools can be developed, but is it possible to moderate this process, this pain gate process, or what happens in the pain gate, without pharmacological interventions? And what we've done in the laboratory is we changed the housing. Normally you have rats housed. This is a normal cage, but you better could say it's a restricted. It's a kind of a jail. It's a very small house for these animals. So they're quite rather depressed. And we housed other animals in an enriched environment with their friends, with toys, with rooms and space and happiness. Of course, we induced a peripheral nerve injury in these animals, housed them immediately in the jail, and others we housed in the enriched environment. And here you see then what we did. Now we make an inflammatory, we evoke an inflammatory response in the peripheral nerve by injection of carrageenan. Here you see again this withdrawal threshold. This is the control pore, the right one, and this one is the one where we injected the carrageenan. You see this becomes more sensitive, as I told you, it now responds to only a few grams and withdraws. Whereas normally you need about 50 grams. And that takes on for 4 weeks. After 4 weeks, we see the injured paw having the same level as the uninjured paw, if housed in the jail. And now we looked in animals housed in the enriched environment. And what we see here is that it only takes 3 weeks before the animals do not feel the pain in their carrageenan injected leg or paw. So there is a reduction of 1 week, which is quite substantial, I think, just by housing these animals in an enriched environment versus a normal, say, restricted environment. And then Anne Gabriel thought, let's go and have a look at this immune response in the spinal doors, in the dorsal horn. Let's look at the astroglial cells, because after 3 to 4 weeks, we know these astroglial cells are activated, they're swollen, they produce this BFGF, interleukin-1B. But what she did is she did a histological study at 3 weeks. In the enriched environment, we know they don't have the pain, whereas in the other animals, they do have. She quantified the immunoreactivity in particular layers, in all these layers, layer 1, 2, 3, 4, 5, and so on, including these 2 and 3 and 4, which are the important pain layers. So again, the housing, and here you see the effect on the glial cells, the intensity of the glial cell as a measure of, let's say, how swollen these glial cells are. And you can see here in the restricted environment, the ipsilateral is still much higher after 3 weeks than the contralateral side for the various laminae. Whereas in the enriched environment, we don't see this difference anymore. So, the astroglial cells, the immune response is slowed down just by the enriched environment. So is it possible to modulate such a very complex interaction in the spinal dorsal horn, which is the basis of the pain response, without using pharmacological interventions? Yes, we are able to do that just by housing. And is this now clinically applicable? Because I told you we have a lot of pharmacological insights, but we're not yet at the moment using this. Yes, it is. Already in 1984, it was published in Nature or in Science that if you house people after a surgery in a room, let's say an enriched room with a view on nature, and you compare them with people who have this view, let's say the more jail-like view, the restricted view, and you then look about the hospital stay and the use of analgesics, it's much less if you have an enriched housing, a healing environment versus a non-healing environment. So that's my last slide, where we focus at the moment on, in our laboratory, what is the effect of the healing environment and treatment of post-operative pain, and the role of the immune response, how we are able to modulate the immune response by optimizing the healing environment. I talked a lot about the central inflammatory response, the neuron-glia interaction in post-operative pain, from acute to chronic. That's it for today. Other questions?